This step-by-step protocol shows how to detect and functionally characterize the enzymatic activity of ring-type E3 ubiquitin ligases, both in vitro and in Planta, through a site-directed mutagenesis approach. By introducing mutation in the ring domain through site-directed mutagenesis, the resulting E3 deficient mutant can be tested in parallel with the wild-type protein to link the enzymatic activity with functionality.
Elucidating the biochemical and mechanical basis of ring-type E3 ubiquitin ligases can contribute greatly to our understanding of their biological significance in development, stress signaling, and maintenance of homeostasis. With slight modification of the in vivo expression system, this protocol can be applied to the analysis of any ring-type E3 ligase regardless of its origin.
Begin by identifying the conserved cis and his amino acids in the ring domain and designing primers that carry the substitution codon of interest flanked by 15 base pairs on either side of the mutation site.
It is critical to properly identify the ring domain, particularly its conserved cysteine and histidine residues. Online tools such as ProCyte can be used to do so.
Use the mutagenic primers and PCR amplification to introduce the desired mutation into the plasmid harboring the gene of interest, making sure to use a high-fidelity DNA polymerase, such as PFU. After PCR, digest the E. coli-derived parental methylated and semi-methylated DNA by adding 3 microliters of DPNL restriction enzyme directly to the